Hello students, welcome to EPG Pathshala program. I am Dr. Shoaib Lukman from the Council of Scientific and Industrial Research, Central Institute of Medicinal and Aromatic Plants, Lucknow. Today we are going to talk on lipid oxidation part 1 from the paper Lipid Metabolism. The objective of the present module includes the understanding of the digestion, absorption and secretion of lipids or fats, and how the dilipidation of lipids or fats takes place. If we see, the main lipids of the body are basically the triglycerides, which are also called as neutral fatty acids, the phospholipids and the sterols, which are the principal and most efficient source of stored energy, and that is because of high fuel value. The 1 gram of fat gives you 9.3 calories of energy. These fats reserves are constantly broken down and resynthesized, and optimal nutrition must contain a certain amount of fat along with the essential fatty acids. Previously, the carbohydrates were attributed for major source of energy, but increasing evidences had established the fact that fat is the chief source of energy for many tissues and perhaps the main source of energy for the body as a whole. In this figure, we can see that how the digestion of dietary lipids takes place in the stomach and has been continued through the addition of lipases in the small intestine. Then finally, bile salts were added and digestive products are formed, and these digestive products include the free fatty acid, which are medium and short chain, the free long chain fatty acid, monoacyl glycerol, cholesterol, etc. Then further, through the action of bile salts and fat soluble vitamins, micelles are formed which facilitate the absorption of the lipids through the intestinal mucosal cells that are enterocytes and assemble with the characteristic protein like FOB48, and finally to form the chylomicrons, which are secreted into the lymphatic systems, which transport them to the various parts of the lipids, the digestion, absorption and secretion. The most important part of the lipid metabolism, how the digestion takes place, after digestion how the lipids or the fat molecule get absorbed, and finally the fate of secretion of the lipid molecule. If we look at the daily intake of lipid, which is approximately 80 grams, of which more than 90% is normally the triacylglycerol or the triglycerides, while the remaining part of the lipids they consist of cholesterol, the cholesterol esters, phospholipids, and unsterilized fatty acid, which is a free fatty acid. And the digestion and emulsification process, which takes place in stomach, is due to the presence of the lipases, the acid stable enzymes. The first of all, lingual lipases, which are present on the back of the tongue, and the gastric lipases, which are present in the gastric mucosa. The lipases mainly digest the triacylglycerol with short or medium chain fatty acids as present in milk. After the breakdown by the lipases, the emulsification of lipids occur in the duodenum, which increases the surface area of the hydrophobic lipid molecules for the effective digestion by the enzymes. And there are two modes of it. The first mode is through the detergent property of the bile salts, and the second method is through mechanical mixing resulting from the peristalsis movement. The bile salts are synthesized in the liver and stored in the gallbladder, are actually derivatives of the cholesterol. The emulsification process actually stabilizes the smaller individual particles and prevent them from coalescing. The dilapidation of lipids, which are the dietary lipids degraded by the enzymes of the pancreas, especially the pancreatic lipases, and the secretions of these pancreatic enzymes are hormonally regulated. The triacylglycerol are large enough to be degraded by mucosal cells of intestinal villi. Thus, they are first converted to the monoacylglycerol and free fatty acid by esterase and pancreatic lipase. In this figure, we can see here that how dietary lipids in form of phospholipid triacylglycerol enters from the mouth into the stomach. This phospholipid or triacylglycerol remains unchanged and directly through the action of bile salt emulsify and digested by the pancreatic lipases. And to form the products that includes the free fatty acid, monoacylglycerol, cholesterol, and remaining pieces of phospholipids. These primary products re-esterified to form the chylomicrons in the lymphatic system, transported to the blood, and thereafter distributed in the different parts of the body. Cholesterol ester. Overall, the dietary cholesterol is present mostly in free, non-esterified form, while only 10 to 15% occurs in the esterified form. Cholesterol ester is degraded by pancreatic cholesterol ester hydrolase, or also known as cholesterol esterase, which produces free fatty acid and cholesterol. This enzyme activity is highly increased in the presence of bile salts. Phospholipids. Phospholipids are acted upon by activated pancreatic phospholipase A2 and ultimately forming fatty acid and lysophospholipid. The lysophospholipid is acted upon by lysophospholipase, leaving a glycerol phosphoryl base that may be excreted in the feces, or it can be further degraded or absorbed by the body. The hormonal control of lipid digestion in the small intestine. The hormonal control includes cholecystokinin, which is a small peptide hormone secreted by the mucosal cells of the lower duodenum and jejunum. This hormone acts on gallbladder, exocrine cells of pancreas, and small intestine. Another hormone, secretin, which is produced by the intestinal cells, helps in balancing the pH for optimal activity. In this picture, you can see how the cholecystokinin in blood through gastric mobility arises, and also after the small intestine, dietary lipids get into the gut endocrine cells and secreted in the blood. Overall, the gallbladder, the cholecystokinin and secretin regulates the digestion in the small intestine. Absorption of the lipids. The absorption of the lipids is mainly done through the enterocytes. These are the mucosal cells of the intestine, and before the absorption process takes place, these lipid moieties are ultimately broken down in the jejunum into free fatty acids, cholesterol, and 2-monoacylglycerol, which are the primary products. After that, micelles are formed in the intestinal lumen, and then these lipid droplets are absorbed in the brush border membrane of the enterocytes. In enterocytes, resynthesis of triacylglycerol and cholesterol esters occurs, whereas in secretion process, the newly resynthesized triacylglycerol and cholesterol esters are very hydrophobic in nature. Therefore, it is necessary to package it as lipid droplet surrounded by the thin layer of phospholipid, apolipoprotein B, and unesterified cholesterol. This helps in the stabilization and increase solubility. These particles, known as chylomicrons, are released from enterocytes into the lacteals, the lymphatic system, present in small intestine villi. Compartments in the utilization of lipids in the animals. If we see the figure, we can assume how the utilization of lipid takes place in the different compartments of the animal system. And these compartments include the lymphatic system, the blood, the muscles, the adipose tissues, the liver, the epithelial cells, and the lumen of the intestine. How this compartmentalization takes place? Let us understand that through diet, lipids gets converted in the lumen of the small intestine into triacylglycerol, and these triacylglycerols, on the action of bile salts and pancreatic lipase, gets transformed into monoacylglycerol and free fatty acids. This conversion then moves to the epithelial cells of the liver, the triglycerides, to form triglycerides plus proteins, plus cholesterol, and plus phospholipids. This all together forms the chylomicron, which is distributed through the lymphatic system and reaches the blood. And from where these chylomicrons get circulated, the free fatty acid gets circulated, the monoacylglycerol gets circulated to the muscles, to the adipose tissues, and gets stored in form of the triacylglycerol or triglycerides. The biosynthesis or the beta oxidation as well as ketone body formation takes place in the liver, again from which the free fatty acid and triacylglycerol is formed, and upon the action of very low density lipoprotein and high density lipoprotein, this moves via blood to the muscles, and where also the beta oxidation gets carried out. Also, in adipose tissue, glycerol 3 phosphates gets converted to free fatty acid and triacylglycerol, as well as to glucose, and which is being transported via the blood to the either to the liver or to the muscles. Use of released chylomicrons by tissues. In skeletal muscle and as adipose tissue mainly, but also heart, lung, kidney and liver, these chylomicrons are broken down to triacylglycerol, which are further degraded into free fatty acid and glycerol by lipoprotein lipase. The deficiency of lipoprotein lipase or its coenzyme apolipoprotein C2, which is also known as familial lipoprotein lipase deficiency or type 1 hyperlipoproteinemia, is a rare autosomal recessive disorder. In this cartoon, we can see that how the amino acid forms apolipoprotein B48, which together with phospholipids enter the chylomicron. Fat soluble enters the chylomicron. 2-monoacylglycerol through the action of acyl-CoA monoglycerol acyltransferase and acyl-CoA diacylglycerol acyltransferase forms the triacylglycerol, which also through the help of chylomicron. Long chain fatty acids, cholesterol, esters also with the aid of chylomicrons transported to the lymphatic system. The destiny of free fatty acid. The free fatty acid may enter the adipocytes or the muscle cells or may be transported to blood in conjugation with serum albumin until taken up by the peripheral tissues. The adipocytes can again produce the triacylglycerols from the free fatty acids, which are stored until needed. In adipocyte, the hormone sensitive lipases hydrolyze the triacylglycerols and releases the fatty acids, which are the free fatty acids, into the blood, where they bind serum albumin, which is a blood protein having a molecular weight of 66,000. About half of the total serum protein, the serum albumins are present, and it non-covalently binds with as many as 10 fatty acids per protein monomer. The insoluble fatty acids have been carried to the tissues such as the skeletal muscle, heart, and renal cortex. In these target tissues, fatty acids dissociate from albumin and are moved by the plasma membrane transporters into the cells to serve as a fuel. Numerous proteins other than serum albumin also act as transporters to carry the absorbed triacylglycerols from the intestine to the different organs and tissues such as muscles, heart, liver, adipose tissues through the blood. These numerous proteins wrap around the lipid molecule to facilitate their movement across the membranes in the cells. In the picture, we can see how these fatty acids with a short and medium chain ranging from carbon 4 to carbon 12 get freely diffused inside the cytoplasm from the plasma membrane. However, long chain fatty acid sometimes needs carrier or active transporter to get it diffused in the cytoplasm. Also, they get diffused to the cytoplasm with the aid of fatty acid binding proteins or some of the fatty acid transporter proteins, which are membrane integral protein or translocases and many other different types of fatty acid transporters. The low molecular weight fatty acid binding proteins, which are called as FABPS, in the cytosol of many animal tissues functions as a carrier of the fatty acids in the cytosolic compartment. The fatty acid binding proteins may also be involved in the cellular uptake of fatty acids, their intracellular storage, or the delivery of fatty acids to the sites of their utilization. The significance of fatty acid binding proteins in the fatty acid metabolism is sustained by the reduction in the uptake and utilization of long chain fatty acids in the knockout mice lacking hard fatty acid binding proteins. These animals exhibit the exercise intolerance and during aging process develop the cardiac hypertrophy. Nonetheless, the molecular mechanism of fatty acid binding proteins functions remains to be elucidated. The destiny of glycerol, actually it is used exclusively by liver to produce glycerol 3 phosphate, which enters either the glycolytic pathway or the gluconeogenesis pathway. About 95% of the biologically available energy of triacylglycerol resides in their long chain fatty acids, and merely 5% is contributed by the glycerol moiety. The glycerol released by lipase action is phosphorylated by the glycerol kinase, and the resulting glycerol 3 phosphate is oxidized to dihydroxyacetone phosphate. The glycolytic enzymes, for example triose phosphate isomerase, convert this compound to glyceraldehyde 3-phosphate, which is further oxidized via glycolysis. See how the destiny of the remaining portion of chylomicrons takes place. After maximum triacylglycerol had been removed, the remaining portion that contains cholesterol esters, phospholipids, fat-soluble vitamins, apolipoprotein, and few triacylglycerols bind to the receptors present on the liver cells and are endocytosed where they are degraded. If this endocytosis is decreased, then they accumulate in the plasma, and the condition is clinically known as type 3 hyperlipoproteinemia or familial beta lipoproteinemia. So students, let us now summarize what we have learned in this module. In this lecture, we have learned about the digestion of the dietary lipids. Foremost takes place in the stomach as well as in intestine. The emulsification process, which is an essential step required for the efficient metabolism of dietary lipids. In milk, the triacylglycerol contain short to medium length fatty acid degraded by lingual and gastric lipase, but other dietary lipids, and they need the pancreatic enzymes. The cells resynthesizes the triacylglycerol, CE as well as PL, and also synthesize proteins, for example apolipoprotein B48, all of which are then assembled with the fat-soluble vitamins into the chylomicrons, which are released in the blood through the lymphatic system. Short and medium chain fatty acids enters the blood directly. Thank you.